Hello friends, welcome to my channel Bio IT Concepts. Today in this video I will tell you how we can design primer for PCR, and if you like this video then please share it and subscribe to my channel. Okay, first of all, the basic info about primer is it is a short stretch of single stranded DNA that is complementary to the DNA sequence of the target gene. Okay, and it is used in polymerase chain reaction. Now, how we can use primer in polymerase chain reaction? We have DNA template 3' to 5' end and 5' to 3' end. Now the primer work in pair. Forward primer here. Here is the forward primer, and if it is 3' then there should be 5'. Okay, and the reverse primer. Here the reverse primer binds. Forward primer moves in this direction and reverse primer moves in this direction. Now next is types of primer. Primer are basically two types, DNA primer and RNA primer. Where we can use DNA primer and where we can use RNA primer. DNA primer is basically used in DNA sequencing and polymerase chain reaction, and RNA primer used in DNA replication. Today in this video I will discuss about the DNA primer and what points you have to remember before designing the primer. Okay, first point is primer length. Second is product length. Third is melting temperature. Fourth is GC%. Fifth is self-complementarity, and sixth is maximum 3' self-complementarity. Now come to the primer designing. In this, we have to retrieve a sequence from the ncbi site. The link of this site is given in the description box. Now, how we can use this site? Here I should type the name of the gene for which we have to design the primer. Suppose I have write the gene ADH. Now we have to design a DNA primer, so I have to select here nucleotide. Now search. Okay, I will take the DNA sequence. Always take FASTA format for primer designing. I will copy this sequence. Okay, copy this sequence, and I am using primer 3 for designing primer. Okay, paste here. Now you remember what are the points I told you earlier? Primer length. Okay, here I will define the primer length. Okay, optimum should be 20, and here I will write 22. Minimum 80 to maximum 22 length. Okay, what should be the TM? Now the TM should be approximately 60 degrees Celsius. Okay, to minimum 57 and maximum 63. Product TM is not required as such. Now the GC%. Okay, GC% always 50. So I will write here 40 and here 60. Now what is self-complementarity? Self-complementarity should be less than 2. Okay, so first of all I will write here 0. If I didn't find any primer with this self-complementarity, then I will increase it up to 2. Okay, now maximum 3' self-complementarity. What does this mean? It Basically, if we put here high value, then there are chances of primer dimer formation, so it should also be between 0 to 3. Okay, now what is the size of the product, or what is the product length? Okay, so I will define here 100 to 300. Okay, now pick primers. So see, there is no primer with this requirement, so I will change the self complementarity by 1. Now I will search again. In this I found no forward and reverse primer. Now I will change the complementarity to 2. Now pick primers. See, now we got both the primer forward and reverse. It is the left primer and it is the right primer. This is the length of our primer. This is the TM. Remember, both TM should be approximately nearby. Okay, GC percent is also okay. Complementarity, 3' complementarity is also okay. Now this is the sequence of primer we get. Okay, this is the product length, and this software is available online. So if you have any queries regarding this, then you can give your comments in the comment box. I will definitely reply. Thank you.